everybody. I am really glad to see everyone today for our Shire's Graduate Seminar Series. And welcome to those of you that are visiting online as well. I'm Jessica Houston, I'm part of the faculty here in chemical engineering. And today I have the great pleasure to introduce Dr. Michelle Farkas. And Michelle is a good friend. I've known her for some years now. And she's actually an associate professor of chemistry at University of Massachusetts Amherst. So I just want to say a little bit about her background first before she shares with us some interesting work she's been doing. So Michelle received a bachelor's degree in chemistry from Wellesley College. Her PhD is also in chemistry from Caltech. She's actually from New Jersey originally. Her PhD work was under the direction of Peter Durbin at Caltech. And after receiving that, she went on to do a postdoc, and her postdoc was at UC Berkeley, and she worked with Professor Matthew Francis on a cancer research project. She was actually a DoD breast cancer research postdoctoral fellow. So Michelle has now been working in an area of chemical biology. She has a lot of interesting projects going on, but she's actually been very productive in her years. In her early years as a junior faculty and now into her associate professor. Some of the awards she's received, she's actually a fellow of the CMC, which is the Chemical Machinery of the Cell. It's a SILOG, and that's sponsored by the Research Corporation of Science Advancement as well as the Gordon and Betty Moore Foundation. So it's nice to be a fellow of that. So she's also was a finalist of the Beckman Young Investigators Program, which is very commendable. And then also she's been doing some work in her department as well on EDI, and so she did receive a commitment to diversity award at UMass. And so I think that's really a great thing and interesting in incorporating that into her day-to-day faculty work in addition to the research she does. So today we're going to hear about her work on macrophages and immune cell, and she's going to be talking to us a little bit about how she tracks them and images them, and help join me in welcoming Michelle.
Thank you for the really nice introduction. Is this on for the YouTube folks over there? Can you guys hear this okay? Great. And I guess for the Zoom folks, that's a separate set of microphones. This is maybe the most technical talk I've ever given. And that being said, I mean, I think we all lived through the pandemic. I'm really excited to be here today because this is actually my first seminar visit since the pandemic. In the fall, I went to my first in-person meeting since the pandemic. You know, it's one of those things we were talking about earlier. You have to physically prepare for days like this where, you know, you're meeting with people all day and you're going to lunch and maybe you get something on your shirt. I'm sorry, Raquel. I'll mention her in a minute, but it's so nice to be here in person and meeting with everybody. I've had the chance to meet with lots of great faculty from both the chemistry and the chemical engineering department. And today's talk, as Professor Houston mentioned, is going to be about macrophages. And so I'll mention some of the other stuff we're doing in my group, and maybe I'll come back another time. But I'm also especially excited to be here because so my wife, Raquel, and her family are from TA, or Tierra Maria, and I'm sure I'm butchering that name 100%. It's up near the Chama Valley. And so my knowledge of New Mexico goes from about this corridor up to Santa Fe, through Española, up here. So it's a very narrow, I wouldn't even call this a rectangle, you know. And so this is the first time that I've ever been south of Albuquerque. And so it's my first time in Las Cruces, it's my first time at NMSU, and so I'm just really excited to be here. But if you ever happen to be up in northern New Mexico, in Chama or something, if you see Tierra wolves, that's my in-laws. So stop in and say hi, and buy something. They're sheep. They're shepherds. They raise sheep, and so it's an amazing area up there. If you've not been, I highly encourage it. So I'm also here on the Chama Valley Tourism Board behalf or something. So on to the science. So I know that this is probably an audience with a pretty broad background. I know that Professor Houston is the kind of the bio-oriented engineer of the department, so I want to make sure that throughout my talk I'm providing enough context for the work that we're doing, which is really steeped in cancer. And so I just wanted to start out with this overview of what happens in a primary tumor, some of the players into distant metastases, which is what really kills patients, is when you have metastases occurring at organs that are vital to life. And so a lot of us think about tumors, we say, oh, we have this cancer cell type. Well, tumors aren't just comprised of a single cell type that might be the MCF7 cells or the MDA and B231 cells that you culture in your lab, but rather they're really heterogeneous and dynamic environments. And the major character that we're going to be talking about today is tumor-associated macrophages. And so one of the things I want to bring up to you all in terms of what you should remember is, one, cancer is dynamic. There are a lot of different cell types. But two, the immune system plays a huge role in cancers as well, right? And so sometimes that role is good, and sometimes that role is bad, and I'll talk a little bit more about that. And then as you go from primary tumors to distant metastases, you have cells that are more resistant to cell death. They're more difficult to kill. They have this enhanced migratory capacity, which is going to come up in my talk as well as invasion. They have their own growth signals, etc. But getting into my research program, so the central theme of this is, Professor Houston mentioned, I'm a chemical biologist. I'm trained as a chemist, but throughout my career, I've been really excited about applying chemistry to solve biological problems. And so actually, before I even went to graduate school, I worked for a pharmaceutical company called Bristol Myers Squibb, and that's where I really got interested in biology. I'll be honest, how many folks here are undergrads? How many graduate students? Okay, cool. So speaking to all the students in the audience here, right? And even the post, student types, because your path is never set. So I took one biology class as an undergrad, and I hated it. It was rote memorization. It was so boring. I never took any more biology as an undergrad. But I really liked making molecules. I loved organic chemistry. I was a chemistry major, and it was only when I went to work for that pharmaceutical company that I realized that, the molecules might be cool looking, the syntheses might be elegant, but it was really about what are the molecules doing in the course of disease. And so I knew that I wanted to have an impact in there. And so when I went back to graduate school, I didn't just study organic synthesis, which was my initial plan, but rather started working in a lab whose molecules were being applied towards answering biological questions. And as I went throughout my training, I learned more about different kinds of biological systems, and I wanted to identify something that would unify chemistry and biology, right? So as I mentioned, biology is dynamic. Cancer is dynamic, right? And so we want to be able to understand these systems, but a lot of the methods that we have are really only taking snapshots, right? So you understand what the system might look like at a singular point in time, right? But you don't know what it looked like before, and you don't know what it's going to look like after. Also, right, as I just mentioned, cancer is complex. There are a lot of different cell types in there, right? And so if you're only looking at one cell type in a really simple environment, how do you know what's actually happening in the broader context of when there are many different cell types, right? And so our basic idea is that we want to look at dynamic systems. So I mentioned that cancer is a dynamic system, biology is a dynamic system, and we want to develop means to track them, right? To follow what is going on in those systems in a temporally and spatially resolved manner, and then use chemistry, small molecules, supramolecular entities, both of which I'll talk a little bit about in this talk, to be able to perturb those systems, to be able to learn more about the systems and how they function, but also hopefully along the way to come up with maybe new treatments, new therapeutic agents, new imaging agents, etc. And so this is the really big picture of my research program. So I mentioned all of biology is dynamic. We had to pick a couple of areas that we were going to focus on. And so we decided to work with two systems that we thought were A, specifically highly dynamic, and B, had not really been looked at in this fashion, right? That were both going to be really important to cancer. So one of them is looking at circadian rhythms. So these are 24-hour biological cycles. I'm not going to be talking about these today. Maybe if I get another invite back, that'll be my next talk. Today I'm going to focus on macrophages, right? So both macrophages and how they interconvert between having these really distinct subtypes and as a result functions, but also using cells as delivery agents. And I'll talk a little bit about why that might be a good approach to use. So as I mentioned earlier, cancer has a really important immune component to it, right? So we can think about all these standard characteristics of cancer. You have tissue invasion and metastasis, insensitivity to growth inhibitors, evasion of apoptosis or programmed cell death. They can develop blood vessels through angiogenesis so that they keep getting nutrients to be able to do this growth, feeding into cancer's limitless replicative potential. And then, again, they can generate their own growth signals. But the part that I'm going to focus on here is the inflammatory environment, right? And so when we think about inflammation, we think about infections, right? So cancer can also be described as a wound that doesn't heal. And macrophages and T cells, although again I'm going to focus on macrophages here, are a cell type that are present in cancers. So just looking at cancers, this is pre-cancer, so no cancer here. We have two tissue types that are being separated by a barrier. We have macrophages already in there. We have blood vessels. Again, they occur naturally in tissues. And then already when you have your primary tumor or your carcinoma in situ, we have tumor-associated macrophages, or those TAMs, right? So from the very beginning, and they're helping the cancer to develop. And so you go towards local invasion, so now you have the cancer going from one tissue type into the other, breaking through that membrane. This is going to be something that's important that we're looking at in some of the assays that we're doing. And you have these cancer cells that are being helped by macrophages to get into these blood vessels to traffic to distant sites, right? And again, you have macrophages there. So to kind of summarize, why do we care about macrophages and their roles in cancer? They promote development of blood vessels in tumors so that tumors can get more nutrients. They can contribute to that tumor environment remodeling so that they can spread through different tissue types and also establish and maintain metastases. They silence the immune response to tumors, so I'll talk about that in a couple of seconds. And then really when it comes down to why we care so much about them is because in most cancer types, and especially in breast cancer, which is our focus, the more macrophages you have in the tumor, the worse the cancer is. The worse the cancer is, the worse the prognosis is, okay? So macrophages, how are they dynamic? Why would we want to be able to track them? And that's because of the fact that their roles are completely different depending on the stimuli that they receive from the local environment. So what does that mean, right? Let's say you're hopefully not in lab, but you scratch yourself, right? You get a cut, you get blood, you have macrophages that are going to come into that wound site. They're going to help clear out the dead and dying debris. They're going to make sure that you don't get any bacteria or anything else coming in to infect it, and that's inflammation, right? You have inflammatory signals, and the macrophages are helping to promote that. But at some point, you need to heal, right? And so those same macrophages are going to be converted from a pro-inflammatory phenotype to an anti-inflammatory or immune-suppressing phenotype. So those macrophages that are helping you heal, what are they doing? They're rearranging that environment that you had that cut in, right? They're helping to develop new blood vessels so that you can get blood into the area where you had the cut, right? And so all of those things that are good for wound healing are also good for cancer. And so we can break down these macrophages into two phenotypes, right? M1, which we call classically activated, and M2, or TAM-like, which are these tumor-associated macrophages that are more commonly associated with wound healing. So M1's high tumor resistance, right? We're thinking about host defense. We're thinking about stimulating the immune system, right? M2, we have low tumor resistance, and these are now thinking about wound healing characteristics, suppressing the immune response, and remodeling that tissue, right? So good for cancer on the right, bad for cancer on the left. And cancers have the ability to recruit macrophages and convert them into these tumor-assisting macrophages, right? So again, the same macrophages can interconvert between one characteristic or one set of functions and another, just depending on the stimuli that's surrounding them. So cancers can take macrophages, they can recruit them, and they can convert them into M2 phenotypes, right? We can have undifferentiated macrophages, and we can also have pro-inflammatory macrophages that will attack the tumors, right? And so one of the questions that we wanted to ask is, how can we study macrophage interconversion and re-education in models of disease, right? Because again, remember, cancer, a lot of different cell types in it, right? Macrophages are going to be responding to different stimuli that's in those environments. So they might respond differently to different cancer cell types. They might respond differently to different chemotherapeutics, right? And we want to be able to track and follow when and how that occurs, including, right, if macrophages are being recruited to the cancer, at what point does the cancer turn the macrophage into a cancer-helping macrophage, right? And so, as I mentioned, a lot of us, including myself and others in the field, have studied macrophages using snapshots, right? Whether doing microscopy experiments, using antibodies, whether Western blots, looking for specific proteins that are upregulated or downregulated, doing RT-PCR, this is an example of, we still do all of these studies in our lab because we want to make sure that the platforms and the systems that we're developing are relevant and we're getting the right readouts, right? And so here we're looking at 4 different markers. 2 of them are associated with the M1 phenotype in green, so again, immune stimulating. 2 of them are associated with the tumor-associated macrophage phenotype, which is immune suppressing, right? And we can see that M1 macrophages have much higher levels of the M1-associated markers, INOS, which is induced nitric oxide synthetase, so that's going to be an important one for the rest of my talk. TNF-alpha, which is tumor necrosis factor alpha, and then for M2s, thinking about things like CD206 and EGR2, which again, upregulated in the M2 phenotype, downregulated in the non-polarized and the M1 phenotypes, okay? So what we did is we decided to make fluorescent reporters, right? So we know that if we have to harvest proteins or harvest mRNA to be able to look at macrophages, we need to have the macrophages by themselves. Otherwise, you're going to harvest all the cells' proteins, right? And harvest all the cells' mRNA. If we did something like an ELISA, where you're looking at what are the cells secreting, right? We're going to be not able to distinguish what are macrophages secreting versus what are the cancer cells secreting. So if we use reporters that are essentially what we're doing is we're taking GFP, which is green fluorescent protein, and we're controlling its production by putting it under the control of the induced nitric oxide synthetase promoter, or INOS, right? So as we said on the previous slide, INOS super upregulated in the M1 macrophages. And so if we can use reporters, now we can take these macrophages that have the reporters in them, and we know A, where the macrophages are, B, how the macrophages are changing in terms of their responses, right? So we can keep following these reporters. We can look at them in M0, M1, and M2 states. We also have the ability to look at things in the single cell level, right? So when you think about Western blot or RT-PCR, you're thinking about averaged distributions, right? If you do something like flow cytometry or you do microscopy reporter assays, we now are able to know, okay, there's actually a pretty heterogeneous population. With something like RT-PCR, again, you don't know how wide a distribution you have among all of your cells, right? Some of the cells that are at the periphery of the tumor might have different iNOS expressions from those at the heart of the tumor, right? But even with no tumor, so this is just cytokine treatment, we know that not all the cells are responding similarly. But we do know that we're able to get a good idea of what's happening and that our reporter has fidelity for iNOS expression. So I mentioned briefly the importance of using different types of cancer models in the sense that cancers have multiple cell types, right? And we talked about how you can do different assays, but for some of those assays, you need to have the macrophages alone, right? Because you can't conflate, okay, what signals are the macrophages giving versus what signals are other cancer cells giving. And because this is a physical science audience, I wanted to make sure that this is clear. So conditioned media is one of the most common ways that we can study cancer, and one of the simplest ways. And we can do this in the sense that we take our cancer cells and we grow them. We grow them in a dish, just like you would any other cell line, and we have media that we're then going to take that's going to have all the components that the cancer cells secrete out. So as I mentioned, cancers are going to secrete different agents that are going to recruit macrophages. They're going to help convert macrophages, possibly into different phenotypes. They're going to have different effects on different cells. So conditioned media, you would take your media that was being grown with your cancer cells, and then just take the media, no cancer cells here, and add that media to your macrophages. And then you look at, okay, what is your macrophage response? You can do conditioned media experiments with not just macrophages, but really any cell type to look at different types of cell-cell communication. And the nice part about this is that you only have the macrophages, right? And so if you take the cell's proteins and look at the protein levels, it's only going to be macrophage-based. If you take the cell's RNA, it's only going to be the macrophages cells. Sorry, only the macrophages material. But there's a lot that's missing here, right? Cancers, again, dynamic environments. There's cell-cell communication and interactions that are happening, right? Not only are the macrophages responding to the cancers are responding to the macrophages, and there's crosstalk where they go back and forth. And we lose all of that crosstalk. There's no crosstalk in the conditioned media. These cancer cells have never seen a macrophage, right? And vice versa. These macrophages are only seeing what the cancer cells were secreting into the media on their own. And so there are two other types of cultures that can be done, but now we get a little bit more complicated because we have multiple cell types growing at the same time. So we can have two-dimensional co-cultures where now we're culturing both the cancer cells and the macrophages together. And we can also have three-dimensional co-cultures. And here's the difference between them, right? So I say 2D versus 3D, great. What does that mean? Why do we care? In 2 dimensions, all of the cells are in a monolayer, right? The cancer cells, when you think about cancer, it's not a flat sheet of cells, it's a clump, right? And so you have a lot of interactions that are happening with those cancer cells. You have packing of cancer cells together, right? And so those cell-cell communications are super important in terms of adherence, but also the environment that each cell is seeing, right? You don't have plastic in your body. When you're growing a tumor in your breast or elsewhere, right, that hard plastic is not going to be the same environment as a soft tissue, right? But by having a 3D spheroid, now you have more of those cancer cells growing in that cluster where you have the cell-cell connections, more of them between the tumor cells or the cancer cells, and you also have more of a relevant matrix that your cancer cells are growing in, right? And so 2D and 3D. And there are a lot of differences that people have seen. When people do drug studies, they've seen that different cancer types respond differently when the cancer cells are cultured in 3D versus 2D, right? There's a lot of biology that's going on in terms of the feedback between the cells and each other and the changes in that environment. And so one of the things that we wanted to do with our reporter first is be able to understand, okay, now that we have our reporter built, we can actually co-culture the cancer cells, but we can also still look at the macrophages and how they respond to just conditioned media and what are the changes between them, right? How much better is this? Are they just going to tell us both the same thing? And the answer is no, they don't. And so I'm not sure how well you can see the color changes here, and so I have these box and whisker plots. Our non-treated, non-polarized macrophages are located over here on the left in gray. And then we have two different cancer types that we're looking at. So these are all mouse cancer because we want to make sure that we keep the species consistent. The macrophages that we're looking at are mouse macrophages, and so we're also looking at mouse cancers. We and others have already published that you can have interactions with human cancers, but we wanted to have the species thread the same. And so the 4T1 cells are extremely aggressive. The EMT6 cells are still pretty aggressive, but definitely not nearly to the same extent. And so when we look at the conditioned media denoted as CM, there is not a very big change between the 4T1 CM and the non-treated cells, right? On the other hand, when you go to the co-culture, these co-cultures are in 2 dimensions, you see that there is a much bigger difference when you're co-culturing the cells versus just with the conditioned media, right? So this is something that you can't see if you didn't have a reporter. At the same time, with these less aggressive cells, it actually, when you just look at the conditioned media, it looks like they're having a greater effect than the more aggressive cells, right? But again, when you go to the co-culture, you see that that's not actually the case. So being able to have these cells together in the same environment is meaningful. Oh, and I should also mention that I have been remiss in updating my website, so I'm getting to it, hopefully in the next few weeks, but if you want to find anything, I don't know if this is being recorded. Maybe it's being recorded, but check back in a month. I'll hopefully have our publication list updated. But this work was just accepted in Frontiers in Oncology, which I found out yesterday. So that's exciting. And then we wanted to look at 3D co-cultures as well. So now we've generated these spheroids. So these are, again, clusters of these cancer cells and looking at the macrophages interacting with them. And we see that there's an even greater extent to which the 4T1, the more aggressive cells, have a greater effect on the macrophages than do the less aggressive cells versus non-treated. So this is pretty exciting because we've been able to show that we can monitor macrophage phenotype. We also see a big difference in the type of culture that you use with the macrophages. So just being able to expose macrophages to conditioned media, which again, a lot of people are doing because it's simple, it's easy, and there are a lot of assays that you can do following that, it's not telling you the whole story. And so where we want to go with this next. So now that we've developed our reporters, one of my colleagues, Shelley Payton, who's in the chemical engineering department at UMass, but luckily we have this setup where we share the same big open lab because we're both in the cancer and biomaterials group. And so her group develops these tissue mimicking hydrogels, and not just of the breast, but of other locations, looking at stiffness and their effects on metastases and drug treatment and things like that. And so now being able to ask the question of, okay, when we have these macrophages in different environments, what are their responses? What do they look like? At what point do we see macrophage interconversion, et cetera? And so doing things like this where we can actually track the macrophages as they enter the spheroids, which is a little bit more intense in terms of the microscopy than some of the initial assays that just my lab had done. I will come back to the reporters and some of the other exciting stuff that we're doing with that, but I want to delve into this part where we're talking about now, we know that cancers can convert macrophages to bad actors, right? But what if we can flip macrophages back, in other words, re-educate them to attack the cancer from within? Right? So macrophages are being recruited to cancers. They're being converted into types that will help the cancer thrive and grow and progress. But what if we can flip that switch and generate inflammatory responses within the tumor so that the macrophages will not only stop that pro-tumor behavior, but also eat the tumor from within. One of the things that I didn't mention to you that maybe I briefly glanced over is that when you have your wound, right, your macrophages are coming in and they're phagocytes. So they're actually eating dead and dying cells. They're actually eating bacteria, right? We can also think about cancer cells being eaten by the macrophages. So one of the things that we've done in our lab, so I do have some slides with chemical structures on them. Not many, because I was thinking about chemical engineers, but I don't think I have any synthesis. I don't know, maybe. I actually might have some catalysis in here later on. My last slide, I think. All right. So we've decided we're not a huge lab. We're not going to compete with pharmaceutical companies, and so we're not doing these huge, big library screens. But what we've done is what I would call a privileged screen, which is where we've taken molecules that have already been identified by others to have specific targets, and we know from the biology end that those targets are involved in macrophage polarization. And so we decided to look at some of these molecules and say, okay, what is their actual effects on macrophages? Can we either have a protective effect where the cancer cells can't repolarize macrophages to the bad phenotype, or can we flip the phenotypes? And so just as one of these examples, we have this one small molecule that we picked up. It was a TLR4 agonist, and this had been looked at in terms of some other models, some antiviral models and things like that, but not actually in terms of what it was doing to macrophages and how we could promote macrophage activation. And so we saw the ideas that we can look at the levels of pro-inflammatory cytokines versus anti-inflammatory cytokines that are being generated, and then look for enhanced phagocytosis and killing of cancer cells. And so these are just some snapshots of some of the data. So here we're looking at TNF-alpha expression. This was before we had the reporters up and running, so this is all RT-PCR and the ELISA assays in this paper. But we can see that we have M1 macrophages, which are pro-inflammatory, M2 macrophages, which are anti-inflammatory. We can treat with this small molecule, which we've termed PBI1, and we can also convert the macrophages after they've been polarized to the M2 phenotype back to an M1-like phenotype with treatment of the small molecule, essentially reversing the macrophage's tumor-associated behaviors. And so, but the real thing that we're looking for is, okay, we can activate these macrophages, but can we get them to eat the cancer cells? And the answer is yes. And so, it's flow cytometry. So we're able to look at the cells, the macrophages eating cancer cells. And so if you look at this bar graph, we have our phagocytosis index. So we have the non-treated cells here, over here at 1. We have an anti-CD47 antibody. And so CD47 is a receptor that's present on the cancer cells, right? So there's a handshake that happens between the macrophages and the cancer cells. Cancer is so smart, okay? So cancers have evolved to be able, or a lot of cancers, I should say, have evolved to be able to display this CD47 receptor that normal cells have on them that tell the macrophages, don't eat me, right? It's called a don't eat me signal. And so macrophages have something called SIRP-alpha. When SIRP-alpha interacts with the CD47 on the cancer cells, the macrophages know not to eat them, right? And so here, with this anti-CD47 antibody, we're blocking the CD47. And so the macrophages, they don't see the don't eat me signal, and so they eat the cells. So that's kind of a positive control. And then we have a couple of phagocytosis inhibitors, cytoclastin D and cytoclastin D plus the anti-CD47, no phagocytosis. And then when we just treat with the small molecule, we actually see similar extents of phagocytosis as we have with the anti-CD47. And even better, so I mentioned CD47 is on the cancer cells. Our small molecule is activating our macrophages. So when we put them both together, we have even greater levels of phagocytosis. And so we decided a few years later to ask the question of, okay, so what is happening in these spheroid models, right? So in this paper back from 2019, we're just talking about 2D cultures, flow cytometry assays, where everything is in liquid suspension. And so we're asking, what is this molecule able to do when we actually have real 3D structures? And so here we have non-treated, so no spheroids, and we know that when we treat with PBI1, we see a pretty big enhancement of fluorescence. And then we know when we have the spheroid co-cultures, right, these cancers are really polarizing those macrophages to a pro-tumor phenotype. But when we treat we treat the spheroid co-culture macrophage model with the small molecules, we can rescue that to some extent. So we can still activate the macrophages in the context of a more realistic tumor environment. And so none of this is at this stage looking at killing or the macrophages invading the cancers itself or phagocytosis, but just tracking. So this was basically where we said, like, okay, let's publish this because we can track the responses of macrophages to therapeutics in disease-relevant environments. And so the next steps are to actually look at, okay, what is happening with these cancer cells, to what extent can we improve killing, but also developing reporters based on other macrophage types and other macrophage markers. So I wanted to include some of our supramolecular chemistry here because I know that that's of interest, broadly speaking, to chemical engineers, even though I'm not sure if there are any supramolecular chemists in the audience here. But we've also done a good bit of work with Vince Ortello's lab, who's in the chemistry department at UMass Amherst, and they are kind of the masters of supramolecular chemistry, anything involving gold. And so this is that interaction that I mentioned earlier, where you have cancer cells that are displaying CD47 and macrophage cells that are displaying SIRP-alpha, right? And so we know that we can treat with antibodies and prevent this interaction from occurring. Well, Vince's lab, the masters of supramolecular chemistry, one of the things that they came up with was basically these nanoassemblies for doing CRISPR. So I'm guessing most of you know that CRISPR is a gene editing technology. And so the idea was that we wanted to be able to deliver gene editing nanoassemblies that would be able to delete the SIRP-alpha in macrophages. So we have these macrophages that have SIRP-alpha, and when we deliver these nanoassemblies, now we have no SIRP-alpha, and so the macrophages can't recognize the CD47 that's located on the cancer cells. And so what we see is that we can track the fluorescence, so we put, in this case, a fluorescent marker into the nanoassemblies as well, and we see enhanced phagocytosis. So these are the normal raw 264.7 macrophages, and these are the macrophages with the SIRP-alpha edited out. And so these macrophages have not been activated even. They're only not able to detect the cancer cells based on this interaction because SIRP-alpha is gone. All right, so I'm going to switch gears a little bit. We've been talking about macrophage interconversion, being able to activate them, being able to track their states. And as I mentioned at the beginning of my talk, macrophages are inherently attracted to cancers. Cancers are secreting chemical entities in the forms of small proteins and cytokine, in terms of aka cytokines, and so cancers can recruit macrophages. And so a lot of people are working in the area of drug delivery, right? Including some of my colleagues, including supramolecular chemists, people working with polymers, people working with nanoparticle-based systems, nanoparticles themselves. And so these have a lot of limitations, right? When I was a postdoc, I was actually working on protein-based systems to be able to do cancer imaging and drug delivery. And in all of these cases, right, you have issues where you don't have enough time in circulation to get to the place that you want to be, which is the cancer. You can have off-target effects, like in a lot of cases, these things will go through the liver or to the kidneys. And again, especially if you're trying to deliver cytotoxic agents, in most cases, let's face it, cancer drugs are cytotoxic, right? And the question becomes, are you going to kill the cancer faster than you're going to kill the patient, right? And so I spent a lot of time banging my head up against the wall, looking at animal images, wondering why wasn't my agent getting to where it wanted to be. And I will also mention the fact that, a lot of times people say that they're working on targeted carriers, right? So if you have what's called a targeted carrier, essentially what you're going to have is some ligand that's going to bind to some protein at the cell surface, and that's the targeting agent, right? I feel like it should have been called something else, like some kind of, molecular glue, although I know that's been taken, for use with something else. But essentially, with those targeted agents, that interaction is only going to happen when your nanoparticle or your drug or anything else is going to get to the tumor. But it's not going to help your agent get to the tumor itself. On the other hand, if we use cells as delivery agents, right, now we have these smart vehicles, or we could have smart vehicles, that are responding to signals that are being sent out into the body by cancers, by diseases like atherosclerosis, infections, right? And so we're not the first people to have thought about using cells as delivery vehicles, for the reasons here. They spend more time in circulation. They're being used, in both tissue regenerating purposes, but also things like blood vessels and loading them. And they're compatible, right? Because they are biological entities to begin with, right? But the difference between what other folks have been doing and what our approach is that we want to decorate the macrophages on the outside, right? So in all these cases, yes, macrophages are being recruited to cancers. But if you have your agent on the inside of the macrophage, you're exposing the cell to your agent and your agent to the cell, right? In a lot of cases, if you have a cytotoxic agent, you're going to kill the cell before you can even get it to be delivered, right? And so you might inject a patient with the cells, maybe 20% of them will still be alive. At the same time, if you have some kind of drug that's in there, that can also be affected by the interior environment of the macrophage, right? We have enzymes, we have lower pHs in some of the compartments. We have issues where loading and release and the control of those two things become problems, because it's hard to control how much of an entity you can get into the cell and how it's released, right? It's just kind of passive release over time. And so our idea was that if we decorate the outside, we can take advantage of chemical functionalities that are inherently present because of the biomolecules that are present at the surface of cells. So I'll talk about that a little bit here. And again, they're being recruited, right, by the cancer, so we're taking advantage of that. So we have a lot of different chemistries that we can employ. We have chemistries that are associated with lipids. We have chemistries that are associated with proteins, right? So thinking about back to biochemistry classes, your amino acid R groups that are have nucleophilic reactivities. We can also install non-native entities, right, like azido groups. I guess it was 22 now, but the 2022 Nobel Prize in Chemistry went to click chemistry, right? And so we can do the same thing so that we can display different types of functionalities. We can selectively do modifications based on the reactive handles that we're attaching to what we want to attach. So we could have, let's say, an imaging agent and a drug, right? Or a chemical sensor and an imaging agent and a drug, or some variation of those. And so when we're thinking about doing now delivery with these cells, as I mentioned, if you have your entities that are inside, you could kill the cell might not even make it to the target, right? And so when we're doing cell modifications, we want to make sure, are the cells still alive? Can the cells still move? Because we want to make sure that the cells can traffic to the disease location. And are the cells still going to be able to respond to the signals that are being given off by those disease environments, okay? So as I mentioned at the very beginning of the talk, thinking about motility, right? Are these cells, once they're modified, going to still be able to traffic? So this isn't following any kind of chemoattraction. This is just looking at migratory capacity. So we have a monolayer of cells in a dish. We induce a wound using a pipette tip, and we watch to see how long it takes for the wound to grow back, or in this case, just looking to see what the differences are between the non-treated and the bulkiest thing that we put on there, which is a giant avidin protein with a fluorophore label. And then looking at the ability of the cells to respond to chemoattractants. So one of the most common chemoattractants that breast cancer secrete is something called colony stimulating factor 1. And so we can essentially do what's called a transwell migration assay, where we have the CSF1 that's located at the bottom of the well. We put the macrophages at the top of the well, and we watch to see how many macrophages actually pass through the filter. And so we can see here that when we have macrophages that we haven't treated with anything, in terms of the chemoattractant, there's nothing at the bottom of the well except for media. We have very little motility. But on the other hand, when we have these non-modified macrophages and we have the CSF1 chemoattractant, we see a lot of migration. But the great thing is that with all of the modifications that we tried on the exterior, we see no statistical change between the modified cells and the non-modified macrophages. So just because we've put a whole bunch of stuff on the outside of the cell means that these cells can still pick up those signals that are being given off. So that's great. We've played around with some extra chemistries. For example, we tried the click chemistry where we installed the non-native azido groups so that we could do the click chemistry. But we realized it takes 72 hours plus to be able to get the internalization of the sugar and the display of the azido group on the exterior of the cells, right? The big picture idea, 20-year plan, is you take a patient's own macrophages, you chemically modify them, and you put them back into the patient, right? Just like CAR T therapy, for those of you folks who have heard about it. So they do it with immune cells, but it's genetic and not chemical. So in this case, we want to chemically modify the cells. And so if you're going to have to wait 72 hours for the cells to take up an agent and then display a functional group on the exterior, that's not going to work. And so we've opted to look at this sialic acid modification where we can do an oxidation. And so we've also, because so many people were asking me about this, what about primary cells? So you keep wanting to say that you're going to do this in a patient, harvest the cells, modify them and put them back in, but you've only ever done this with immortalized macrophages, which are more robust. I won't go into immortalization and what the difference is between primary cells and immortalized cells, except to say that primary cells are the cells that are directly coming from an organism, right? So when I say bone marrow-derived macrophages, these cells actually came from mice, right? These cells, basically came from a giant cooler, and they went through a whole bunch of things, and they came from mice in the way back when, but not recently. So looking at this, we see that, despite most people's inclinations, including my own, that the primary cells were going to be more sensitive and delicate, they weren't. One of the things I was surprised by is that the primary cells overall migrated less towards the chemoattractants, but when you look at the differences between the non-treated primary cells and the treated primary cells versus all of the chemically modified cells, we see relatively little to no difference when we modify the cells. So even with the primary cells, this works. And this talk is about macrophages, but we've also been using stem cells because stem cells are another cell type that a lot of people have been using as vehicles, and it works. How am I on time? Okay, great. I can do this in a couple more minutes. So we're obviously interested in cancer. We want to use these things as delivery agents, but we also are still interested in questions like the extent to which macrophages will associate with different types of cancers. And strikingly enough, and we still haven't really looked at this either, is that we know that the more macrophages people have, the worse the prognosis is. We know that there are different breast cancer types out there, and some of them are more aggressive and some of them are less aggressive, but nobody's actually looked at the extent to which amounts of macrophages correlates with cancer type, right? And this is really something that you'd want to do in a human, system, and we don't do anything with patients or patient-derived samples. But at the cellular level, and this was published back in 2018, we've been able to show using these modified macrophages and 3 different breast cancer types, so MCF7, our luminal A, SKABR3, our HER2 positive, and MDAB231 are triple negative, and triple negative is widely considered the worst type of cancer that you can get. It's extremely aggressive. It affects young people, disparately affects African Americans. And so from left to right, you can consider this as least aggressive to most aggressive. And if you look at this, these macrophages are literally coming in and they're interacting with and almost picking up those cancer cells, right? And so, I'm not going to draw any more conclusions, but to say that, it looks like there's a greater extent of interactions between the cancer cells and the macrophages in the more aggressive cells. And so, again, wanting to do this as delivery agents, this is the only ex vivo image I'm going to show. I'm not going to show any live images. But essentially, we want to make sure that we can get to the tumors. And in fact, so this is all ex vivo using an IVIS fluorescent imager. We can see that we have the agents at the tumor. And over time, as the agents get secreted through the liver, et cetera, they will stay in the tumor. And one thing I haven't mentioned with regard to standard delivery agents versus what we're doing with the cells is the fact that tumors have necrotic environments, and these are often really hard to reach for those agents. For a lot of those agents, it's hard to get too much outside of the blood vessels or even outside of the periphery of the tumor. But macrophages are also attracted to those necrotic interiors, and so if we cut one of these tumors open, we can actually see that there's a really nice segment of macrophages in there. So there's a lot that we have left to do with this, and this is just kind of a little bit of like where we're going right now. So in terms of drug delivery, the big difference between imaging and delivery is that you need to be able to release your drug, right? So we need to be able to disconnect the drug from the cell carrier. And so we're working on developing cleavable linkers that are also going to give us an extra element of specificity for the tumor environments, because those linkers are going to be susceptible to cleavage based on the characteristics that are in the tumor. So we're talking about lower pH, we're talking about higher levels of specific proteins called matrix metalloproteinases, and we have a number of other things that we can also deal with, like looking at oxidative environments and things like that. But right now we're starting with pH and peptide-based linkers based on matrix metalloproteinase cleavage sites. And then the other great thing that I really very briefly mentioned is that if we're decorating the cells on the exterior, we can also develop sensing agents, right? And so one of the big areas that people really don't understand is, what is the difference between tumor environments from one tumor type to another? I'm thinking in terms of pH, in terms of the presence of different reactive species. Like a lot of people are developing sensors for things like hypoxia or hydrogen sulfide. And so, but we don't have the means to get into the tumor to be able to really probe what do things look like at different parts of the tumor, right? And so in these cases, having a cell-based delivery vehicle where these sensors on the exterior is needed, because otherwise, if the sensor is inside, all you can see is what is the environment like on the inside of the macrophage. And I should have mentioned this. So I have 2 graduate students working on this, but we are exclusively working on the cell-based decoration. The sensors are coming from other groups, some collaborators of mine at SMU and Illinois. So the last thing I'm going to mention here, and look, I have a little inorganic chemistry. So this is again collaboration with Vince's group, and so we are not prejudiced against loading things on the inside of macrophages. And there is, I believe that there's a time and a place for everything, and the perfect place for this is to load supramolecular structures where you actually don't want the structure to come out of the macrophage, right? So there's an area of supramolecular chemistry referred to as nanozymes, right? And the idea behind these entities is that they are carrying out chemical reactions that can't be done just exposed to biological entities because they're toxic, right? So if we look at this catalyst, right, we have an iron palladium catalyst system, and so the idea is that we can embed this in this supramolecular gold nanoparticle based what we call nanozyme, and then we can put this nanozyme into the macrophage cells. And so what this will allow us to do is, I mentioned before that most agents for cancer treatment are severely toxic, right? But we can do what's called a prodrug strategy. And so what a prodrug is that it's an inert version of a molecule, right? And then some chemical reaction has to happen in order to liberate that protecting group so that you'll have the active version of the molecule. So I have two versions here. One is fluorophore, so this is rhodamine. So pro-rhodamine, we have these propargyl groups here. And then upon exposure to the catalyst, we have liberation and cleavage of these groups, and then we have just the rhodamine. Sorry, it's not just the propargyl. I realize that it's this entire amide we then cleave, and then we have the amine. Similarly, 5-fluorouracil is an extremely toxic chemotherapeutic. And so we could deliver this prodrug, which has no toxicity, and convert it upon interaction with this catalyst into 5-fluorouracil at the reactive entity, right? And so now we're putting together the specificity of the macrophages. So if these macrophages are loaded with the nanozyme, right, we know that the macrophages are going to migrate to the tumor, and so this catalyst is not going to be exposed to the biological environment, and you're also only going to convert the 5-fluorouracil from the inactive version to the active version in the tumor, because that's where the macrophages are, right? So what the entire body is going to see is this inert prodrug, right? We're only going to be able to do the conversion in the macrophages. And we've also actually, this is not cancer related, but we've also done this using a salmonella model in macrophages as well. But there we're actually trying to treat salmonella in the macrophages. And so again, we see with the nanozyme, we have no change in the cell viability to the macrophages because we're not affecting them at all. We see that even once we've loaded the nanozymes into the macrophages, we don't affect their ability to migrate to the tumor and the chemoattractant. And we see that, the prodrug itself, no effect. Just having the macrophage loaded with the nanozyme, no effect. And when we have them both together, we see results that are essentially almost similar to the drug itself. So the drug itself is in gray, and our co-culture is the HeLa cervical cancer cell line with the full macrophage nanozyme system is very active. So that's the end of my talk. I realize that I went a little bit over from what I was supposed to or thinking about doing, but hopefully I've been able to tell you guys about macrophages and why they're interesting and some of the things that we can do to be able to understand their behavior, regulate their behavior, and take advantage of their behavior as well. And so the last slide here, this was the group last spring. A few of these folks have graduated. This is like the post-COVID graduation, trend or something. A lot of people had to not take time off. It wasn't time off. It was forced time away from the lab. But Joe was the joint student between myself and Vince. He actually graduated just before COVID, so it's like, phew, thanks. Javier just graduated last summer. He did the work with the macrophage reporters. Vishnu, Kyle, and Jeffrey have been working with cell-based delivery. And before I lose my voice, I'm going to say thank you to everybody, and thank you everybody online, and I'll take any questions, or at least as many as we have time for. All right, thank you.
Time for a couple. And if you're online, you can enter it into the chat.
I have a question. Yeah, so you mentioned knocking out SERP alpha entirely on macrophages, and obviously you're getting increased phagocytosis of those cells. So obviously SERP alpha isn't required for that recognition event. Do you happen to know any of the mechanisms of recognition of macrophages to cancer cells that don't require SERP alpha? Because if you just knock it down entirely, it seems like you are losing a certain degree of control.
So you're right? Because if we are knocking down SERP, or knocking out, in this case, SERP alpha, we lose the ability for the macrophages to recognize, that signal, which is not just given by cancer cells, but that's given by normal cells. So cancer cells have kind of adapted that to say, no, we're normal. It's kind of like a cellular disguise. And so, in that case, and also in the macrophage activation case, in any of these instances where we're trying to activate macrophages, we are always also thinking about off-target effects, right? Because we don't want to phagocytose normal cells. We don't want to activate macrophages that are everywhere in the system, right? And so in those cases, so for example, the cell-based editing that I was showing, was kind of a proof of concept study, but really what we want to do is we want to edit those macrophages in the tumor environment, right? So we've played around a little bit with our collaborator in terms of intratumoral injections. We've also dealt with some things in terms of, how do we just get that system to the macrophages that are resident in the tumor, or have the system turned off until or unless the macrophages are in the tumor. Because again, we don't want to activate macrophages, throughout the body, and we also don't want to edit the macrophages to not be able to realize that a normal cell is a normal cell. The great question.
Thank you for your nice, good presentation. I just wonder, how did you isolate 3 different of macrophages when you want to study their markers at first?
So
Are they primary from patients?
So great question, and I should have clarified this. So any place that I said raw 264.7 macrophages, those are immortalized cells. So those are cells that have been originally derived from an animal, but then processed in different ways, and we can talk about that offline. But essentially, so their original lineage is the same as what was isolated from the animals, but those changes that have occurred have also granted them different behaviors, right, and different characteristics. And so I mentioned in the case where I was talking about the cell-based delivery agents, how we did it with the immortalized raw cells and the primary cells, right, because there have been changes to those raw cells where they're model cells, right, they're still representative of macrophages, but they're not as close to the real thing as what's directly from animals. So in the primary cells, those cells were isolated from mice. What we do is we usually use bone marrow-derived cells, and so they're progenitor cells, and so they're stem cells, and we differentiate them into macrophage cells. And then what we do is use them for our assays. So when you're talking about having the different phenotypes, so in all of these cases, we have macrophages, and then we can use cytokines, so we can basically reproduce what's happening in the body. So there are known agents that are known to stimulate macrophages to go to one phenotype or another phenotype, and we can treat the macrophages that we've either isolated or taken out of the freezer with those cytokines to be able to generate those phenotypes.
And I have the other question about this Pro-5FU that you use for treatment. You injected this Pro-5FU transform like or transfected macrophages to the tumor environment?
So those studies, and that's actually published work in JAX AU, their open access version, so anybody can look at that. But essentially, so we're looking at that in a tumor model, but not in animals. But the idea and the reason why we want to do this is because we know that Pro-5FU is non-toxic, right? And so if we have this macrophage nanozyme composite only in the tumors, we'll only generate this 5FU at the tumor site, right? But these studies, and this is the citation here, these studies were not done in animals, but that would be the idea. And that's what we're getting towards, but haven't done it yet. Yeah.
Running up on time, so I just want to say that Dr. Farkas will be in the conference room if anybody has any questions they want to talk to her about. So, but join me in thanking her for a great seminar.
Thanks, everyone. And I guess graduate students and undergraduates can join at that time, so just feel, even faculty, anybody.
Thank you, everyone.
Thanks all.